Welcome back to my channel. Today I will discuss about enzyme part 2. Let's talk about the classification of enzyme. On the basis of catalytical behavior, we can classify enzyme on 6 types. These are oxidoreductases, transferases, hydrolases, Lyases, Isomerases, Ligases, and recently added Translocases. Let's dig into details. First, oxidoreductase. From the name you can easily identify that this is related to oxidation and reduction. This is actually transfer of electron, catalyze oxidation to one substance with reduction of another substance. That is, what is oxidation and reduction? As you know, oxidation is the addition of oxygen or removal of hydrogen or removal of electron, and the opposite is reduction. That is loss of oxygen, addition of hydrogen, and gain of electron. In this reaction, for example, A is containing H and plus B, both are substrate. Here A became oxidation. Why? Because it lost hydrogen, and B gain it, so it's reduction. So that's way oxidation and reduction happen. And the example is lactate dehydrogenase, succinate dehydrogenase, and catalase. This is another example how oxidation and reduction occur. Here loss of hydrogen, that is oxidation. Here gain of hydrogen in case of bromine, so it will be reduction. And there is an example of lactate reductase, which EC value 1.1.1.27. EC value 1 means it's class 1, first class oxidoreductase. Here if you look closely in lactate structure, you see here OH and here hydrogen, that means hydrogen. Here hydrogen, here also hydrogen, and here this lactate is substrate and NAD cofactor, and here pyruvate is product. In case of pyruvate, loss of hydrogen. Here loss of hydrogen and it form pyruvate, and here NAD gain hydrogen, so it's reduction. So this is an example of oxidoreductase class of enzyme. Let's talk about transferases. From the name transferases, it actually transfer a group but not hydrogen, bear in mind. That means if A and B, they are substrate or substrate cofactor, something like that, and here transfer of a group from B to A, here A gain a group and B lost it, so here transfer of group. And what type of group transfer? Acyl group, alkyl group or amino group or phosphate group and glycosyl group. And the example of this group is kinase, aminotransferases or transaminase. Here for example hexokinase, you find it in glycolysis reaction where glucose transferred into glucose 6-phosphate. Here glucose is substrate and ATP, from ATP phosphate group is added with the glucose, so it forms glucose 6-phosphate. Bear in mind, usually phosphate group when transfer, it's kinase type. So this one is hexokinase. Why hexo? Because of having glucose here, this glucose 6-phosphate. And another example is L-alanine and alpha ketoglutarate. Here alanine transaminase. Transaminase means here an amine group is transferred. How it transferred? From alanine, this amino group transferred and it formed into pyruvate, loss of amino group, and gain this alpha ketoglutarate gain amino group and it become alpha glutamate. So here the enzyme is alanine transaminase. Let's talk about hydrolase. Hydrolase, from the name you can easily identify. Hydro means water, that means split of water. So here enzymes that bring about hydrolysis, that means water lysis. The breaking single bonds, here single bond will be broken by adding water to hydrolase ester. It can hydrolyze ester bond, peptide bond or some other bond, and most of the digestive enzyme are in this group. Which digestive group? Like lipase, sucrase, or others. And here, for example, AB, this is the bond, and this bond break by here by hydrogen and OH, this is from oxygen, and then H came here and OH came here. That's how bond are broken. Example is triglycerol. Here lipase, how lipase act? Lipase break here in ester bond. Here in ester bond broken by using lipase enzyme, and then here it add OH group and here it add H group. That means it produce diacyl glycerol and fatty acid. This is an example of hydrolases class of enzyme. Let's talk about Lyases. Lyases is actually breaks C and C bond, that is carbon-carbon bond, carbon-oxygen bond, and carbon-nitrogen bond. In Lyases, in this group of enzymes, they can break this carbon-carbon bond, this single bond, but other than hydrolysis or oxidation. And again, they can add double bond, that is the addition of double bond or formation of double bond. And the example is Aldolases. Aldolases, they actually cleave C and C bonds in sugar. For example, fructose 1,6-bisphosphate aldolase in glycolysis, and decarboxylase, they remove a carboxyl group, that is pyruvate decarboxylases, and synthesis. Bear in mind, this is synthesis. And in another group, that is synthetase, it's in group 6. So here synthase means they can form without using ATP, and in group 6 synthetase that need ATP, that is the in group ligase group. Here is an example, AB. Here they break this A and X bond, B and X bond with the help of Lyases, and then they form double bond, and this XY also bond, this form single bond. And again in reversible reaction, that is they use the synthase, they can also form without help of ATP. Let's look an example, pyruvate decarboxylase. As I told earlier, decarboxylase remove carboxyl group. Here pyruvate decarboxylase works on the substrate pyruvate. This one is pyruvate, and they cleave in this point and then remove of this point, that is the remove of carboxyl group, and make the carbon dioxide and also acetaldehyde. That's how Lyases work, and they form double bond. What about Lyases in EC group number 4? Just I showed in this in another example. In case of aldolase, as I told earlier, aldolase work on group fructose 1,6-bisphosphate. You will find it in glycolysis. Fructose 1,6-bisphosphate, here carbon-carbon bond breakdown. And this form glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. Here aldolase or fructose 1,6-phosphate aldolase work on this substance fructose 1,6-bisphosphate and cleave it into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Let's talk about isomerase. Isomerase EC group number 5. They convert a molecule from one isomer to another. That's why this is called isomerase, and that is the transfer of a molecule within a group. Bear in mind, it will be within a group to yield a isomeric condition. And they also help to facilitate intermolecular rearrangement. Intermolecular rearrangement is important. Intermolecular rearrangement in which bonds are broken and also formed. So 3 things. First one, they convert a molecule from one isomer to another, and they transfer a molecule within a group to yield isomeric form, and also they facilitate to intermediate rearrangement. How they did it? They broken or formed. Like all type of recimerases and epimerases are in this group, isomerase group. For example, phosphoglucomutase, triphosphate isomerase, phosphohexose isomerase. These are the example of isomerase group. Let's have a look on the example. For example, triose phosphate isomerase. Triose phosphate isomerase work on dihydroxyacetone phosphate. This is also again in glycolysis, and then they just shift their group to form dihydroxyacetone to glyceraldehyde 3-phosphate. Here you have seen the change of the group, but everything is same. And another is glucose 6-phosphate, which is an aldose, and it form fructose 6-phosphate by the activity of phosphoglucoisomerase. Here also 6 carbon compound glucose 6-phosphate, which convert into fructose 6-phosphate. Same, everything is same, just rearrangement among the molecules, intermolecule. Let's talk about ligases. Another name of ligases is synthetases. As I told earlier, that synthetases use ATP to bond to form the bond. This is the formation of bond, that is carbon-carbon bond, carbon-sulfur bond, or carbon-nitrogen bond by condensation reaction. Bear in mind, it will be condensation reaction, and two molecules join by the using of ATP. So here ATP is used. For example, AB to substrate, they bind together to form AB by using a molecule of phosphate, that is by using ATP, and it form ADP. And example is acetyl CoA carboxylase, DNA ligase, glutamine synthetase, bear in mind, not synthetase. So here the example, like acetyl CoA, this one is acetyl CoA and bicarbonate. So by using ATP, acetyl CoA carboxylase add acetyl CoA bicarbohydrate with acetyl CoA and form malonyl CoA, and also it convert in ADP. Let's talk about translocases. Translocases is the 7th group, newly added enzyme. They helps to catalyze movements of ion or molecules across membrane. For example, proton pump, acyl carnitine translocase, or acyl CoA synthetase. These are the example of translocases. This is the movement of ions or molecules across membrane, and this is the general reaction. For example, from site 1B will transfer in site 2B, and this is the product. So that's how translocases work. Let's talk about the summary of this section. On the basis of the reaction, we can classify enzyme in 7 groups. These are the EC number 1, EC number 2, 3, 4, and 5, 6, and 7. First one is oxidoreductase, transfer of electron, that is the hydride ions or atoms ion, accepted or donate. And then transferases, here the transferases group of transfer of reaction. They can transfer acyl group or amide group or ammonia or others. And hydrolysis reaction, that is transfer of functional group to water by adding water. And Lyases, addition of groups to double bond, they form double bond. And also sometimes removal of group or breakdown of bonds like CC bonds or CO bond or CN bond, but not hydrogen bond. And isomerase, transfer of group within molecules or intermolecular chains to yield isomeric form. And ligases is the formation, or this is the joining of two molecules or two substrate, like formation of CC bond, CS bond, CO bond, and CN bond by condensation reaction, bear in mind, and coupled with ATP cleavage. And final, that means use of ATP. Another name of ligases is synthetase. And translocases, this one is catalyze movement of ions or molecules across membrane. It helps to catalyze the ions to pass through the membrane, like proton pump or acyl carnitine translocases. Let's talk about enzyme kinetics. Enzyme kinetics, what it means? It means quantitative measurements of the rate of enzyme catalyzed by reaction. That is quantitative measurement. And here the most important proposal is Brown proposal. What is Brown proposal? Overall reaction composed in two elementary reaction, and that is the substrate, and this one is enzyme, this one substrate. So they form a complex which is called substrate enzyme complex, and then they decompose to product and enzyme, then they decompose to product and enzyme. That means enzyme and substrate, they make a complex which is intermediate condition, and then product produced and enzyme released. So here K1, this one we can call K1, and this one is reversible minus K1, and this two is K2. So in equation, that is Michaelis-Menten equation, they consider this K2 as constant, and this is negative K1, and this one is positive K1. Is forward and reversible, but this one is irreversible, bear in mind. Let's talk about the steady state kinetics. That is, this one is substrate and enzyme. Substrate and enzyme, by the time being, if time increasing, then substrate decreasing, bear in mind. If time increasing, substrate decreasing, but product increasing. If time goes, product increasing. And what about enzyme substrate complex? It remain constant. For the very beginning it may be changed, but most of the time it remain constant. That is the basic principle. Let's talk about Michaelis-Menten model or kinetics. In Michaelis-Menten model, here if we say in x-axis substrate and y-axis is the velocity, initial reaction rate, and when in the very early stage where substrate are free, so velocity will increase, and the time being it will increase. At the one level it will be maximum, which we called V max. That is the substrate concentration at which enzyme shows maximum activity. That means in this state enzyme showed maximum activity, and here also enzyme is saturated. That means so far substrate that will be react with enzyme are saturated. That's why this is the maximum, and after that the velocity will be decreased because there is no substrate remain to bind with the enzyme. And in 1913, Michaelis and Menten they introduced this equation, which this equation is V equal V max substrate divided by Km plus substrate. What V means? V means velocity of reaction, V max is maximum rate achieved by the system, and S is concentration of substrate, and then Km stands for Michaelis constant, I will show it, and again substrate concentration is S. Here in graph, as I told earlier, in substrate and rate, in the time being, the reaction substrate when it goes, and also velocity will increase. At one level it will be the maximum, after that it will again decrease. So in this we called Vmax, and the half of this we called Km. Half of this is Km, bear in mind. So what is Km? Km is the concentration of substrate which permit the enzyme to achieve half Vmax. So half Vmax is Km. Non-Michaelis-Menten enzyme. Bear in mind, model or Michaelis-Menten model, they actually deal with one substrate, one enzyme, but it's very hard to measure ES complex. This is the limitation of Menten model. And what about if enzyme and substrate, enzyme has more than one substrate, then what will happen? That can explain in non-MM enzymes, that is non-Michaelis Menten enzymes. Here they call by reaction. This can be occur in two way. One is sequential, ordered sequential, and also product also release sequential. In this concept, actually substrate bind in specific order. Look at here, this one is substrate A, this one substrate B. Enzyme first bind with in substrate A, that is EA, and then bind with substrate B, that is EAB. And then what will happen? Then the both substrate bind, and then products will released, and product will release in specific order. Look at here. Here product is say EPQ, then P will be released, and then after then Q will be released, and then enzyme will be free. This is the order sequential for both substrate and products. And sometimes it also happen that random sequential, that means binding of substrate and release of product is not ordered. That can also happen. Let's talk about Lineweaver Burk plot, which is called in 1934 it was introduced, and which is called double reciprocal of equation. In Lineweaver Burk plot, this is the Lineweaver Burk plot. How it achieved? Let's see. Here, as I told earlier, this is the equation of model, that is Michaelis-Menten model. Here V0 velocity is equal Vmax substrate, Vmax multiply substrate divided by Km plus substrate, and if we reciprocal it, 1 by V0 equal, it will be from down it will be up, and then if we again separate it like this way, Km by Vmax plus substrate by Vmax by substrate, and then it will be like a linear model. 1 by V0 equal Y, and then M stands for Vm by Vmax, which is M, and then for 1 by S, that is 1 by substrate equal X, plus 1 by Vmax equal C. Look at here, in the y-axis it will be 1 by V0, in x-axis it will be 1 by S, and in M it will be, that is the M, it will be Km by Vmax. This is called Lineweaver Burk plot or direciprocal plot. Let's talk about the inhibition of enzyme activity. Why enzyme needs to be inhibited sometimes? To regulate enzyme activity, to activate it or to inhibit it on the basis of the biochemical reaction. This is very important, and our different pathways need this. Let's talk about the inhibitor. What is inhibitor? Molecules that interact with enzyme and that of course inhibit the reaction. It can be done by temporary or permanent, and it actually reduces the velocity of the reaction or prevent enzyme to work in a normal manner. That's the main function. Let's see here, this substrate and enzyme, this is active site, and what will happen? Substrate bind with enzyme in active site, and then product release, and then enzyme also release. This is the normal procedure. And what happen if inhibitor can block here, and substrate can't came contact here, and then there will be no reaction. This is called inhibitor. And inhibitor, we can classify inhibitor in two way. One is reversible, another one is irreversible inhibition. In reversible inhibition, we can also classify it into competitive inhibitor, non-competitive, and uncompetitive. Let's dig into here. Competitive inhibitor. Competitive, from the name you can easily identify, it compete with the substrate, that means it mimic the substrate but do not undergo the chemical reaction. It only mimic, and it also bind the active site. Look at here, this is the substrate, and this here is the active site and enzyme. And what actually competitive inhibitor do? They mimic as like the substrate, and then they block the active site. Bear in mind, competitive inhibitor always bind in the active site, not allosteric site. And then what will happen? Then substrate can't bind with the enzyme and no reaction will be proceed. And the example of reversible or non-covalent bond inhibitor in Krebs cycle succinate anhydrase. This one is succinate anhydrase, succinate dehydrogenase, succinate dehydrogenase converts succinate to fumarate. Here succinate is the substrate and then it convert it into the product fumarate. And what will happen when this succinate is mimicked by malonate, which is look exactly like the succinate substrate, then succinate dehydrogenase can't produce any product. That means no reaction. This is the example of competitive inhibitor. Let's talk about non-competitive inhibitors. Non-competitive inhibitors, it bind to the allosteric site. Look at here, this is the active site and this one is allosteric site. So non-competitive inhibitor bind in the allosteric site, and then what happened? Look closely that this active site conformation is changes. Now it little bit changes the active site conformation. Then what happened? Then substrate can't bind with this enzyme because of the change in the conformation. This is called non-competitive inhibitors. So the basic difference of competitive and non-competitive inhibitors is non-competitive inhibitor always bind with the allosteric site, not the active site. For example, heavy metal ions, silver, mercury, lead, they inhibit the activity of variety enzyme like urease, for example, and also cyanide and hydrogen sulfide. They also inhibit the activity of catalase and peroxidase. As I told earlier, competitive they bind in the active site and non-competitive they bind in different site, that means allosteric site. Let's talk about uncompetitive inhibitors. Uncompetitive inhibitors are those that do not bind to free enzyme but they bind to enzyme substrate complex. That means this inhibitor bind in the enzyme substrate complex. Then what happened? Then cause the change of the enzyme shape. This change the active site enzyme active site conformational change, and so that what happened in this altered active site substrate can't bind properly, and so no reaction proceed or no production find. For example, hydrazine inhibit aryl sulfatase, phenylalanine inhibit alkaline phosphatase. These are the example of uncompetitive inhibitors. So what's the basic difference between non-competitive and uncompetitive? Non-competitive, it bind allosteric site, it's okay, but it's attack on enzyme. But uncompetitive, it attacks on or it bind with enzyme substrate complex. Let's talk about irreversible inhibitor. What is irreversible inhibitor? Irreversible inhibitor forms a covalent bond. They form actually a covalent bond with an amino acid side chain in the enzyme's active site. That means they make a covalent bond in the amino acid side chain in the active site. Of course, what it happen? It causes the substrate to be excluded. That is substrate excluded from the active site, and it's a permanent inhibition. That's why it's called irreversible inhibitor. Like iodoacetamide irreversibly inhibited the catalytical activity of subenzyme by modifying cysteine and other side chain. That means it's modified cysteine and other side chain. Let's talk about rate limiting enzymes. What rate limiting enzymes are? Rate limiting enzymes are the slowest enzyme in a metabolic pathway which determine the overall rate of the pathway. That means overall rate of the pathway is determined by some slowest enzyme. That means their activity is the very slowest, and it also called committed step enzyme or key regulatory enzyme. These are the rate limiting enzymes, other names also. because they are usually the main regulatory point. As I told earlier, they will be the slowest enzyme in any metabolic pathway, and they are the main regulatory points. That means they control the by allosteric regulation, and most of the time rate limiting enzymes is usually the target of feedback regulation. Usually they are the targeted of feedback regulation. I will talk details about feedback regulation in next slide. Let us know about some rate limiting enzymes. For example, in glycolysis, phosphofructokinase is the rate limiting enzymes, and also in glycogenesis, glycogen synthetase, and also like HMP shunt, glucose 6 phosphate dehydrogenase, TCA cycle, isocitrate dehydrogenase, and urea synthesis, CPS1 carbamyl phosphate synthase. They are the rate limiting enzyme, and there are so many rate limiting enzyme in different pathways. Let's talk about the feedback regulation. What is feedback inhibition or feedback regulation? These are the regulatory mechanism in which the end product of a metabolic pathway inhibit the activity of the enzyme earlier in this pathway. That means if you look at this figure, A produce B, from B C, and from C D produced, and then after producing this D, it block the activity of the enzyme E1. This enzyme is responsible to produce B from A, so it block this enzyme's activity. This is called feedback inhibition. That means the final product of this metabolic pathway inhibit the activity of enzymes of this earlier pathway, the same pathway in the earlier enzyme. Usually this blocks the rate limiting enzymes, as I told earlier, usually but not always, bear in mind. They are targeting rate limiting enzymes which are the slowest enzyme activity in any pathway, metabolic pathway, and they are the key enzymes which control the whole pathway. These are the rate limiting enzymes. So feedback inhibition always target these rate limiting enzymes, not always, most of the time, and this feedback inhibition prevents unnecessary buildup of the product. For example, ATP inhibits phosphofructokinase, that is the end product feedback. In product feedback, ATP produce, when ATP produce in high energy, in high level, then it inhibit the activity of phosphofructokinase 1. And what's the key feature of feedback inhibition? This type of regulation is allosteric. That means the end product bind to the site other than the active site, and this is reversible. This is reversible means when the end product level decrease, then inhibition is lifted. That means it can regulate it based on the amount. If it's lower, if it's getting lower, then the feedback inhibition will switch on. That means it need more product, and vice versa. Let's see how activation and inhibition occur in enzymes. For example, in a high affinity form of an enzyme, which is high affinity, the allosteric site this one, active site this one, and what will happen? Substrate bind in the active site, and then product will produced and enzyme released, and then this high affinity form of enzyme is blocked by allosteric inhibition, or maybe in other inhibition may occur like competitive, non-competitive, irreversible, or feedback inhibition. This can occur, and then what will happen? This will little or no product formation, which you call low affinity form of an enzyme. That means here no product will form, or very limited product will be formed. And what about the activation? Activation means turning enzymes on. This is the allosteric site and active site, which is little or no product formation, which we called low affinity form of enzymes. Then allosteric activator bind here, and then they become high affinity form of an enzyme. Then substrate will bind with this enzyme and product will release. And how this activation occur? It can occur by adding cofactors or allosteric activators or proteolysis or covalent modifications. By this way, a low affinity form of enzyme can be turned into high affinity form of a enzyme. Summary. Let's have a summary of the whole lectures. Enzyme kinetics. Enzyme kinetics is the quantitative measurement of the rates of enzyme catalyzed by reactions, and enzyme rate depends on substrate concentration until saturation, which we have shown that Michaelis Menten equation, V equal V max substrate divided by Km plus substrate, where V equal initial reaction velocity rate and Vmax is the maximum velocity when the enzyme is saturated with substrate, and substrate concentration is S, and Km is the Michaelis constant. And then we can see enzyme inhibition. Enzyme inhibition is when a molecules or an inhibitor decrease or prevent the activity of an enzyme based on the need of the reaction or pathway. And then this one is enzyme inhibitors. Enzyme inhibitors we can classify in two ways. One is reversible, that is competitive, non-competitive or uncompetitive, and another one is irreversible, that is the covalent or permanent binding. And rate limiting enzyme, the slowest enzyme of any pathway, which is the regulatory bottleneck, we can call it, because it will control the whole metabolic pathway. For example, phosphofructokinase in glycolysis. And feedback inhibition, when the end product of a metabolic pathway inhibit earlier enzyme, that is usually the rate limiting enzyme they target to prevent overproduction, known as feedback inhibition. Thank you very much for watching.